Okay, hello everybody. So this is the last lecture from me. And what we need to do is finish up with bacteriophage, then have a little look in about the last hour, I guess, about general features of viral infections in humans and a little look at SARS-CoV-2 and COVID-19. Not forgetting the most important thing in the whole course, which is microbe of the week, of course. So it's the last, so that we'll get the results of microbe of the week at the end of the class, okay? So hopefully it will all fit in the time, but I happen to know that Madame Morato is going to be here with you straight afterwards. So if we run over a little bit, I'm sure it will not be a big problem, okay? All right, so last time we left with these images of a chlorella virus infecting a cell and the different stages in the infectious process that can be visualized by electron microscopy. And the kind of experiments done in bacteriophage and done on viruses infecting cells and looking at what was happening at different times led to the basic model of the replication cycle, which is true for all viruses, okay? So that's what all viruses have got to find a way to perform each of these steps. So first, viruses have got to bind onto the host cell. They've got to find a way to get inside the host cell. There's no point in hanging around outside. And at some point, the virus genome has got to be released from the capsid. So this step is called decapsidation. Now in many viruses, entry and decapsidation occur at the same time. But for a lot of viruses, first the particle is internalized and then in a subsequent step, the genome is released. So once the genome is released into the infected cell, that's when you have the eclipse phase. At this point in time, after decapsidation, there's no more infectious virus particle. All you have is an infected cell. And this viral genome has got to accomplish two things. First, viral genes have got to be expressed to produce virus proteins. And the genome has got to find a way to be replicated. It replicates itself. So the infected cell accumulates a large number of new viral genomes and a lot of virus proteins. And then the structural viral proteins will associate with the new genome copies. So new particles will be assembled. So after this assembly stage, then you can start to detect infectious virus particles inside an infected cell again. And, of course, there's no point hanging around inside this infected cell. In order to go and infect new target cells, all viruses have got to be released. And this has got to be, it's got to be an active process. Okay, it's not passive. And so one of the things that this means is that if you can block any one of these steps, then you can prevent the virus infection from spreading in an organism, okay? So if you block attachment or entry, so this is how antibodies work, okay? They'll bind to the surface of the virus and prevent these early steps in the infection process. So blocking attachment and entry, you stop this cycle from beginning. There are many viruses can be treated, viral infections in humans, with antiviral drugs. And these drugs will mostly target these two steps, okay, the replication of the genome or gene expression. And there are some antiviral drugs which can inhibit this release step. So, if you block one step, then the whole thing can't be completed. And of course, what's going to be very effective is if you combine different therapeutic approaches, blocking several steps in the process, and that will make it very difficult for a virus to replicate. Okay, so we're going to have a look at how bacteriophage accomplish these different stages. And the first step in the process is to bind onto the right target cell. Now, this is very important, especially if you think about bacteriophage in the marine environment. They're like released from an infected cell, they're free in a liquid medium, and there's a whole bunch of different organisms around there. So if you're a bacteriophage that likes to infect particular species of bacterium, there's no point binding to the surface of some seaweed, okay, because you can't infect that type of organism. You've got to bind to the right cell. And this specific binding is mediated by the interaction between an attachment protein or on the phage surface and a receptor on the host bacterium. So I've just noted down here a few of the attachment proteins on different types of bacteriophage. And for the tailed phages, Myoviridae, Cephuviridae and Podoviridae, all these attachment proteins are down here at the base of the tail. Okay, that's where they are. So the phage is going to attach onto the target cell down at this end of the virus particle. Now, the receptors, what are they? Well, they're obviously some kind of structure that's on the outside of the bacterial cell. Okay, so phage T4, M13, phage lambda, they all infect Escherichia coli, gram-negative bacterium. So in gram-negatives, you have the outer membrane, right? And phage have got to bind onto something that's accessible. So that's why one of the receptors here is OMP-C, outer membrane protein C. It's a porin in the outer membrane of the bacterium. Bacteriophage M13 binds onto the F pilus. Okay, so this is a big, long structure, the conjugation pilus on the outside of the bacterium. Now, I wish I should have taken my pens here. So if you visualize the F pilus, right? Everybody got it in their mind. You've got the bacterium here, and you've got a big filament outside the bacterium. Now, if the phage binds at the top end of the F pilus, that's going to be a problem for infecting the cell, right? Because, okay, so here's your bacterium, here's your F pilus. If the phage binds up here, it's going to be no good, right? Because it's got to get inside the cell somehow. So M13 actually has got the ability, after phage binding, it induces the retraction of the F pilus. So after the phage binds, somehow it induces the retraction of the conjugation pilus. So as this pilus is re-internalized into the cell, it brings the phage in there as well. Okay, so that's a pretty clever way to get inside the cell. Lambda phage, the receptor is LAMB. This is a permease for maltose. Okay, so that's another outer membrane protein which is accessible. And a lot of bacteriophage infecting gram-negative bacteria also bind to LPS O repeats on the outer membrane. So LPS is often a receptor. So this is one of the functions of the capsule around bacteria, because these outer membrane proteins, LPS antigens, can be targets for phage that infect bacteria. So bacteria that have a capsule limit the access to these receptors. Okay, so that's one of the reasons why the capsules exist. Okay, so in gram-positive bacteria like Bacillus subtilis, the cell wall has a very different structure. So the types of receptor proteins that phage use are going to be very different. So this, I only picked out one example here. UEB is a membrane protein, but it's very large and it spans the peptidoglycan layer. So there's a part of this protein which is accessible on the outside of the cell wall. Okay, different receptors, they've got to be on the outside of the cell, and so they're different between gram-positives and gram-negative bacteria. Okay, so that's attachment. Then how do bacteriophage get into cells, or at least transport their genome into the infected cell? So this is very well understood for several phages, particularly the T-even phages like bacteriophage T4. And so the first step is binding to the receptor, and this will induce conformational change. Wow. So, I don't know, the hydrophobic domains here in the base plate will traverse the outer membrane, and then there are several copies of lysozyme which are attached to this base plate, and they'll be released and degrade the peptidoglycan layer. So the phage punches a hole through the peptidoglycan, and then the tail contracts, and then this will form a tube that crosses the plasma membrane. And then finally, the DNA of the phage is injected into the cytosol. So these are the steps for bacteriophage T4. Similar thing for all the T-even phages. So the bacteriophage T2 was used by Hershey and Chase in this famous experiment to show that DNA and not protein was the genetic material. So of course, in this case, entry and decapsidation are happening at the same time. Okay. So that's my wonderful PowerPoint animation that I created. Oh man, so long ago now, I don't even want to think about it. But there is better video of this process. Oh, no, come back. Don't go away. Up here. Yeah, turn the sound off. So there you go, you've got your T4 phage. And all the structures are accurate. Okay, so it's a different color for different proteins or different structures on the phage. So it's binding to OMP-C, then the base plate is kind of deployed, hydrophobic domains go across the outer membrane, and you get these phage lysozymes that are released and pierce a hole in the peptidoglycan layer. Okay, so peptidoglycan is broken down, and then the tail just contracts and forms a tube that crosses the plasma membrane. Okay, so what enters into an infected cell is the phage DNA. And once the phage genome is injected, the first thing that will happen is that it's going to circularize, in fact, and then the different genes, the different promoter regions on the phage DNA can be activated to start transcription of the early messenger RNAs. Okay, so not all of the phage genes are going to be transcribed at the same time. Initially, 2 minutes after infection, you just get transcription of the early genes. And these early genes are going to do several things. Firstly, degrade the host DNA with a phage DNase. So that's one of the early genes. And then the phage will start to replicate its own DNA by expressing a DNA polymerase. Now, can anyone see a problem with this process? We have two things going on at the same time which shouldn't happen. Everybody get this? So phage DNA polymerase, it's producing DNA, right? DNase is going to be degrading DNA. So how can this actually work? Because normally what would happen is if the phage is making new DNA with this polymerase, it should all be degraded. But there's a way around this, and the way around it is that bacteriophage T4 doesn't use the same chemical structure of the cytosine base as normal DNA. It expresses another early enzyme, which is hydroxymethylcytosine synthase, which will modify cytosine that's released as it degrades the host DNA. And it's this modified base which is incorporated into the phage DNA. And the enzyme will only cut the host DNA. It won't cut DNA that contains hydroxymethylcytosine. So this is a clever system for the phage to discriminate between the host cell DNA, all gets degraded, and new phage DNA, which can't be degraded by the phage DNAs. So it's really necessary for this enzyme to be expressed at the same time as the DNA polymerase. Okay, once DNA replication has occurred, then, let's bring this back a step. So after DNA replication, then you get the expression of the late genes. And the late genes are expressed from promoters that can only be transcribed in the presence of this phage protein, GP55. And GP55 is a sigma protein. Okay, in molecular biology, have you done this before? Have you done this yet? Bacterial RNA polymerase. No? The core RNA polymerase in bacteria is a tetramer, alpha 2, beta prime. Yeah? Ringing some bells there. And this cannot bind to DNA unless it's associated with a sigma factor, right? This is what binds to the promoter. So GP55, this is a sigma protein. So in the infected cells, you get this complex, and this is what targets the RNA polymerase to the late gene promoters. Okay, so that means you can't get expression of these late genes until you've already got GP55. So what do the late genes code for? Well, they code for the structural proteins of the phage. So the head and the tails will be made separately. DNA will be packaged, and then the heads and the tails will be assembled together. And then right at the end of the process, you have to have release. Okay, so this is the gene replication, genome expression, assembly. The last phase, the last step in the cycle is release. And bacteriophage T4 actively lyses the cell. Okay, it will produce lysozyme at the end of the cycle and a protein which has a name called the holin, which will make holes in the plasma membrane. Because if the lysozyme is expressed in the cytosol, it can't get access to peptidoglycan, right? So it needs a pore protein to allow it to come through. That's the holin. Okay, so that's how phage T4 replicates. So it's a typical lytic cycle. Early genes expressed, which allow phage replication. Late genes, DNA replication. Late genes are expressed are structural genes and the genes that allow release of the bacteriophage. And of course, it's very important that these genes are expressed at the right time, because if the phage starts to express lysozyme and the holin right at the beginning, of course the infected cell would explode before it had any infectious particles produced. So the timing is really important. Okay, I'll come back to this later. Let's do the question a bit later, because now it's just what I've just said. We'll see if you remember. Okay, so that's a lytic cycle. Now, not all bacteriophage perform a lytic cycle. So typically, the big classic for bacteriophage that is not always lytic, is bacteriophage lambda. So bacteriophage lambda, it's named after Esther Lederberg, who is a microbiologist who discovered this. And what occurs sometimes with a bacteriophage lambda infection is that the infected bacterium can still survive, doesn't get killed. And you can see this in this video microscopy I'm going to show you, which was from a paper about 15 years ago now, where they genetically modified phage lambda to express the green fluorescent protein GFP, so that you can see which bacteria are infected. Okay, so here's a start. Oh, you've got two infected bacteria. Wow, one of them explodes and the other one doesn't explode. Okay, the other one carries on dividing. So that means after the infection, you have two very different fates for these infected cells. At the beginning, you've got this cell, it's infected, and you also have this cell, which is infected. This one is going to be killed. This one survives. So what is happening in these two cases? Well, for this cell, what is happening is that phage lambda is conducting a lytic cycle, just the same as what we saw for bacteriophage T4. So the first thing that happens is the phage injects its DNA into the cell. This DNA will circularize, phage genes get expressed, the DNA is replicated, you get a lot of copies of DNA, it's packaged into new particles, and then they're released. So we have a lytic cycle. However, in this cell, something different happens. The first steps are the same. So you get the phage, which will bind to the outside of the bacterium. The phage DNA is going to be injected. It will circularize. And then what happens is that instead of replicating a large number of copies of phage DNA, it's going to be integrated into the host chromosome. So once you have bacteriophage DNA inserted into the host chromosome, this is what's called a prophage. And only some of the phage genes will be expressed from a prophage, and none of the genes involved in phage DNA replication, particle assembly, or release. So all these late stages of the phage lytic cycle are suppressed. So what that means is that this cell, which has been infected, it's got a prophage in its genome, expressing, in our case, the green fluorescent protein. This bacterium, well, it just carries on living, right? And if it's growing, then it will divide. And every time this bacterium divides, it's going to copy the prophage along with the rest of the chromosome. So two very different things happen here. Now, this process, when you have integration of a phage genome into a bacterial chromosome, is called lysogeny. Okay, so this is a lytic cycle and this is the lysogenic cycle. And it's a cycle because this prophage can sometimes be reactivated. So if the prophage is reactivated, then it will be excised from the bacterial chromosome. You get the expression of these late genes, and then the bacterium that was carrying the prophage will be killed, and you get a lytic cycle starting up again. So bacteriophage, like lambda, will be switching from one mode of replication to another, a lytic cycle and a lysogenic cycle, and back again. Okay, so one of the questions is what decides the fate of the infected bacterium? The initial stage is identical. You start with phage DNA injected into the bacterium. Now, what makes the decision between a lytic and a lysogenic cycle? are the relative levels of these two gene products, C2 and CRO. So C2 and CRO are the first two phage genes to be expressed after a lambda phage infection. And what is going to happen is if you have a large amount of CRO, then, or an excess of the CRO protein, then the phage will go into a lytic cycle. If you have an excess of the C2 protein, then the phage will go into a lysogenic cycle and not kill the bacterium. Now, in a lot of cases, well, the C2 protein is often degraded by host cell proteases. So what are the conditions that favor the accumulation of C2? Well, one of them is low temperature. So at a lower temperature, the bacterial proteases aren't very active and C2 can accumulate, and then this will push the phage into a lysogenic cycle. Another condition is if you've got a low metabolic activity in the host cell. That is, if bacteria are not in the exponential growth phase, if they're in the stationary phase, this will favor the accumulation of C2 and lysogeny. And the other thing that can happen is if you've got a lot of bacteriophage infecting the same bacterium at the same time. So there'll be more copies of the C2 gene and you get more expression of the C2 protein, and this will favor lysogeny. So that's how it's induced. In terms of molecules, what are important are, for example, an integrase. So after the expression of C2 directs the expression of an integrase, which will integrate the phage genome into the host chromosome, and it will also activate the expression of the C1 repressor protein. And this will turn off the genes involved in lytic replication. So that's very important. So these two proteins are essential for initiation and maintenance of lysogeny. Now, yeah, so this is a kind of complicated process, right? And everything that is complicated in biology happens for a reason. Because if it's really complicated and it's pointless, then it will disappear. You'll have by natural selection. So this is a kind of genetic program that's been maintained, so it must be worth something for the bacteriophage. Now, what seems to be kind of the characteristic of the conditions that favor lysogeny is that these are not good conditions for bacterial growth, okay? Now, and that kind of makes sense because if you're a bacteriophage and you're infecting bacteria that are growing exponentially, that's a good time to have a lytic cycle because you can be sure there are going to be a lot more uninfected bacteria around than you'll be able to infect them afterwards. But if your bacteria are not growing very well, or if you've already got a lot of free bacteriophage hanging around, you've got more phage than host cells, there's no point in having another round of lytic infection because the phage that will be released won't find good bacteria to infect, okay? So that's really the strategy behind lysogeny. So if the conditions are not favorable for bacterial growth, or if there's already too many phage around, it's better to induce lysogeny and try and wait for a later time to restart a lytic infection. So in order for this to make sense, then the lytic cycle, it has to be possible for the phage, for the prophage, to be reactivated, okay? Because there's no point integrating into the bacterial chromosome forever. That's it, your existence as a virus is finished if you do that. So lysogeny has got to be reversible. And this reactivation of the lytic cycle is provoked by things that are stressful for the host bacterium. So it can be a heat shock, ultraviolet radiation was the big classical one, also some antibiotics. So if this bacterium that's carrying the host prophage can become stressed, so it might be killed, right? Then the prophage kind of realizes, oh no, my host is going to die. I better get out of here. So it's going to reactivate a lytic cycle and produce a load more copies and release phage that can go and infect other bacteria. And that kind of also makes sense as well. There's no point sitting around in a host that's going to be killed. So the lysogenic cycle makes sense for the bacteriophage. It's optimizing the replication strategy. Okay, so yeah, just a couple of reminders of terms. Phage DNA that's integrated is called prophage, and lysogenic phages are also called temperate phages. Temperate means they're, you know, quite soft, not nasty. Okay, so I'll go back to this question here, see what you remember about phage T4. Oh, it doesn't let me go back to this slide, weirdly enough. Ooh, yeah. So have a think about these questions and I'll activate them on Meadow. Okay, where is it? Where is it? Yeah, this one. That's it. It's totally my last question. Okay, that's it. It's been activated. So let's see what you remember about this. I think there should be more than one correct answer. Okay, I can see what you've sent in. So mostly good. Okay, starts with the interaction between the attachment protein on the phage and bacterial receptor. Correct, that's true. 2, afterwards, phage enters a bacterium. False, it's false, right? It's just the DNA, not the whole phage, just the DNA enters. Early genes are involved in lysis. No, false, it's late genes. GP55 is essential for the expression of late genes. True, that's true. And this one, 5 is also true. Not many people got this one. Because remember, the initial step, you have the phage lysozyme punches a hole in the peptidoglycan to allow the tail to punch through. And at the end, phage lysozyme allows the release of T4 phage from the infected cell. Okay, so we get up to here. So that's about it for bacteriophage replication. You know, these are incredibly classic examples. Everybody should know these. Okay, T4, lytic infection, lambda lysogeny. And I think, you know, for me, what is important is to understand why this lysogenic cycle is useful for bacteriophage. That's why it exists. Okay, so last thing to say about phage before I go on to like human viral infections is, you know, what's the impact of phage on human health? There's mostly negative, I have to say. So the negative side is that many temperate phages carry virulence factors. and they are one of the mechanisms by which bacteria can exchange virulence factors. So all of these pathogenic bacteria, some of their important virulence factors are carried on prophage, okay, often exotoxins. And this is one that you will have seen already in the TD, enterohemolytic Escherichia coli is extremely dangerous because it has these two prophage that allow the bacterium to express these Shiga toxins. Now, of course, these prophage don't just sit around in the same bacterium. Of course, they can transfer these virulence genes between bacteria by transduction. And in fact, this gene transfer is possible between different species of bacteria. And this is one of the main mechanisms by which harmless commensal bacteria can become pathogenic. They can acquire virulence factors from pathogenic bacteria. So this is an important impact of bacteriophage on human health. Now, the positive side, I guess, is the potential use of bacteriophage as antibacterial agents. And this was really first thought of by Félix Derel, who popularized and in fact commercialized several bacteriophage as treatments for bacterial infections. Now, this kind of was left to one side after the discovery of antibiotics, but the growing problem of multi-resistant, antibiotic-resistant bacteria has meant that there's been a bit of a renewal of interest in bacteriophage. Now, one of the big places which is famous for phage therapy is the Elyava Institute in Georgia, which of course used to be part of the Soviet Union. And they didn't really have access to a lot of antibiotics during the 1940s, 50s, and 60s. So they maintained and grew up their collection of bacteriophage, which were used in the clinic. And they still do use them. And rather unusually, there are or were some centers in the States who also use bacteriophage for treating, for example, burns or wounds on the outside. So that kind of makes sense, right? Because if you've got an infected sore on the outside of your body, you can put the bacteriophage right on the wound and hope that the phage will infect the bacteria that are contaminating the wound. So they published some impressive successes. Now, what has been kind of new, so this was a little bit of an anecdote, but over the last 10 years, there have been some really amazing stories of, it's just isolated case reports of people who basically would otherwise be dead, but who've been treated with bacteriophage therapy. So in 2016, there's someone who had a very, very serious infection, a 68-year-old man. Acinetobacter baumannii is a kind of opportunistic gram-negative pathogen. And this person, he was really almost dying, and he was in a coma practically. And they gave him a cocktail of 9 lytic phages, and then he managed to eradicate the bacterial infection, and he survived and was discharged from hospital. Now, another case report 5 years ago now, another elderly patient, another gram-negative bacterial infection, resistant to antibiotics. And this one's really clever because they gave him a dose of a particular bacteriophage plus an antibiotic. And that worked. Now, what is clever about it is that the phage receptor here for the OMK01 phage is an efflux pump. So this is a membrane protein in Pseudomonas which exports antibiotics. So it's one of the mechanisms of antibiotic resistance in this bacterium. So really, it kind of trapped the bacterium between two things. If it's going to become resistant to the phage, it could reduce the expression of the receptor, but if it reduces the expression of the receptor, it becomes more susceptible to an antibiotic. So this was a really clever combination of antibiotics and phage to eliminate a particular bacterium. So the person who does a lot of research on this is a guy called Paul Turner in Yale, who's really doing great stuff. So if you have a chance to check out Paul Turner, ask him for a lab internship next summer. That would be good. Okay, so new interesting bacteriophage. They've got some advantages and some drawbacks. So one of the things about them is they're highly specific. So bacteriophage will only infect a certain number of host bacteria. So the positive point about that is that it won't disturb your normal microbiota, unlike antibiotics. But the drawback is that that means they're very narrow spectrum. So you have to match every infected bacterium, every bacterium that is in the person to its own particular phage. One of the things that's good about them is that what you want to use are lytic phages. You don't want to use lysogenic phages, lytic phages. And what is great is that every lytic cycle, you get more of the phage in the person. So this is kind of like magic. You're giving some kind of drug and it will produce itself inside the person you're treating. So that's really great. And the other good thing about it is that the mechanisms for killing infected cells, they're nothing to do with antibiotic resistance. Okay, so antibiotic resistant bacteria will still be susceptible to phage. Now the negative point here is that if you kill and lyse all the bacteria that are inside an infected patient, then perhaps you can liberate endotoxins and you might have a toxic shock. So this is perhaps a consideration, so maybe there's some drawback here. And the other thing is, okay, as a drug, it's a bit unusual because it's actually a virus particle made out of protein. The advantage is it's not going to be toxic. You're not going to have chemical toxicity, but it can be immunogenic. So if you are injecting these phage into somebody, that person will generate antibodies against those phage proteins. So that means probably you can only use them like once. You can't like have a repeated therapy of the same phage. And they don't last very long in the blood. They're cleared by your immune system. Okay, so I think that's about it for phage. And I'm not going to dwell on this. I'm going to go straight to the last lecture, which is about viruses, human viral infections, and SARS-CoV-2. Okay, so first I'm going to go over some general features of viral infections. And most of the time when you talk about viral infections, people automatically think about something like this, you know, I wasn't feeling too great last week, I had a virus. So what that means is you get infected, the virus is going to replicate, then you'll have some symptoms and your immune system will kick in and you eliminate the virus. That's it. You've had an acute viral infection. And if you catch an adenoviral infection or rhinovirus, then that's about it. You get a cold, then you get better. Now, of course, there are some acute viral infections that are extremely pathogenic, like Ebola virus, rabies virus. Ebola virus, if you catch it, as I said last time, you've got about a 50-50, or Zaire Ebola virus, I should say. There are 3 different species. So Zaire Ebola virus, if you catch it, you've got about a 50-50 chance living or dying. And that's if you get therapy. If you have a rabies virus infection, well, if you don't get any treatment, you're definitely going to die. It's like 100% case fatality ratio. And in between these 2 extremes, you have other things like influenza, SARS, or COVID-19, where, you know, not everybody recovers. Some people have a very severe form of the infection and some people die. And as well as that, some viral infections are persistent. They last several years or even decades. And there are 2 types of persistent viral infection. So one of them is, or one type, are latent infections. So in a latent infection, you get a primary infection, virus replicates, it can be found in the blood, you have symptoms, then it looks like everything it looks like everything's fine. You can't find virus in the blood anymore. There's no active virus replication, but it's still there in some cells in your body in a latent form. And these latent forms can be reactivated from time to time. So you'll get reactivation of viral replication, and this will give you reappearance of the symptoms. And these cycles of latency and reactivation, they go on for perhaps almost the whole of your life. Now, as well as that, there are chronic viral infections. So here the difference is, during a chronic infection, there is always active virus replication. And in that case, the replication can bring on symptoms or can be asymptomatic. But as time goes on, the tissue where the virus is replicating will become more and more damaged. And the symptoms have a tendency to get more and more severe as time goes on. And it can lead you to die from a chronic infection. So, for example, the herpes viruses, herpes simplex virus, Epstein-Barr virus, they are the classic latent viruses. You catch them when you're a child, and you'll probably be latently infected for the rest of your life. Okay, you don't get rid of these viruses. Chronic viruses are the hepatitis viruses, hepatitis B, hepatitis C. So they go on replicating in your liver for forever, and if you don't get antiviral treatment, hepatitis C will probably kill you. That's not entirely true. The process is very slow, probably takes several decades to get cirrhosis and liver cancer. So if you've got chronic hepatitis C, you've got a 50-50 chance of dying of hepatocellular carcinoma, and the other 50% is just heart attack, get run over by a car, just like everyone else, okay? During the intervening time. And in the middle, you've got other things that are kind of slightly latent, slightly chronic, like HIV. Okay, so those are viral infections. And of course, to infect somebody, every virus has got to get inside the body somehow. So mostly it's mucosal epithelia that are the sites of infection. So, gastrointestinal, respiratory, or genital. Skin is not really a good site for infections, for viruses, because on the outside of our skin, this is all dead cells, right? So if a virus arrives there, it can't infect a live cell. So most of the viruses that start their infection at the cutaneous site are transmitted by mosquitoes or ticks. So these arthropods will bite you, and the virus is injected under the skin, and that's how they meet their first infected cells. Now, there are some viruses that can enter just by a little scratch like that on the surface of your skin. So papilloma viruses that cause warts on your skin, the verrucas, levion, that's the kind of thing where they're infecting directly at the skin. Blood, can be a source of viral infection. Blood transfusion, intravenous drug use, if you're using injected material and you share it, that can transmit viruses from one person to another. Another one, a bit unusual, organ transplant. If you receive an organ transplant, sometimes you can get viruses, especially these latent viruses that stay somewhere in the infected person. And last but not least, very, very important, is mother-to-child transmission. Okay, so this is huge thing for hepatitis B, for example. For HIV is very important, and this occurs either in utero during the pregnancy, but it can also occur during birth or after birth with milk, with breastfeeding. So some viruses can be excreted in breast milk and can infect infants. So these are the ways that viruses can be transmitted, and it's very important to know what transmission route is used, because that's what determines what is going to be effective to control an epidemic. Okay, so for COVID-19, wearing a condom is not going to help. okay? Totally different transmission mechanism. So that's why these things are important here. Okay, wearing a mask is not going to protect you from dengue fever. Okay, so that's why this is important. Okay, once you get infected, what's going to happen? Well, some viral infections stay localized, and that means that they just replicate at the initial site of the infection. So the virus can't be found in the blood, and these generally have a short incubation time. Incubation time is the time between infection and symptoms. So rhinovirus causes a common cold, okay, infects you up here, stays up there. Rotavirus, these infect your gut, and they just replicate in the gut. They don't go in the blood. Papillomavirus, they infect you on the skin, they stay on the skin. Coronavirus, same kind of thing. Infect the respiratory epithelium, they stay there. Well, maybe in the severe forms they have a bit more systemic. So other viruses cause a systemic infection. So that means that the target organ, the organ that's going to be damaged that gives you symptoms, is not the same as the initial site of infection. And so to get from one place to the other, the virus has got to go through the blood system at some point. So you always have a viremic phase in a systemic infection. So these infections tend to have a longer incubation period. After you're infected, it takes longer for symptoms to appear. And typical example, HIV, okay, infection site, genital mucosa, target organ, lymphoid organs, goes through the blood to get there. Measles, you might not think it, okay, so the site of the infection is, you breathe it in, it's respiratory virus. The symptoms are on the skin, right? All these little spots on the skin. To go from here to all the way around your body, it's a systemic infection. HAV is hepatitis A virus. So it's going to infect you at the gastrointestinal, in the gut, and the target organ is the liver, so it's got to go through the blood to get there. Okay, so that's enough generalities. I've got about 20 minutes to talk about coronaviruses. So I'm afraid it's going to have to be quite quick. So one thing everybody should know is that there are 4 different species of seasonal endemic coronaviruses that infect humans. And then there are these new coronaviruses, the MERS, SARS, and SARS-CoV-2. And by sequence homology, they can be divided into different genera, okay? Alpha coronavirus is a genus of the family Coronaviridae. Okay, so there are, among the endemic coronaviruses, there are 2 alpha coronaviruses and 2 beta coronaviruses. And these new ones, they're beta coronaviruses. Now, one thing to remember is when you say COVID-19, you're talking about the disease. When you say SARS-CoV-2, that's the virus, like AIDS, HIV. So what does a coronavirus look like? Well, it's like this, an envelope virus with a helical nucleocapsid with a genome which is positive strand RNA. The virus particle is kind of simple. There are 4 structural virus proteins. The spike glycoprotein, the nucleocapsid protein around the RNA genome on the inside, and in the membrane there are 2 proteins, M and E. So what's going to be important for us, really, for the rest of our consideration, is the S protein, because the S protein is the virus attachment glycoprotein. Now, the basic replication cycle for RNA viruses all happens in the cytoplasm of the infected cell. So after entry, you have the RNA genome which is released. Host cell ribosomes will start expressing viral proteins. Among those viral proteins, you have at least an RNA-dependent RNA polymerase, which will allow the replication of the virus genome. And then once you've got enough genome copies and viral proteins, they assemble and they're released. Now, have a little bit of a closer look at this. So what are the receptors for these different coronaviruses? So SARS-CoV-2 has this protein, the ACE2 protein, which is the same receptor as SARS-CoV-1. Now, you might think, oh, well, that's why it's so pathogenic, that's why it's so dangerous. But that's not true. That can't be true because one of the endemic seasonal coronaviruses also uses the same receptor. So that's not really the reason why it's so, these SARS coronaviruses have been so problematic. So for endemic coronaviruses, the entry mechanism seems to be that after binding to the receptor, the particle's internalized, then the S-glycoprotein will be digested partially by proteases in the endolysosome, and this is what will allow the fusion of the host cell membrane and the envelope of the virus, which will allow the nuclear capsid to get into the cytoplasm. So I think I'll show you this video. We'll probably still have time. So that's for the classical endemic viruses. However, for SARS coronavirus and SARS-CoV-2, this membrane fusion step occurs at the surface of the cell, which I think is what we'll see here. Yeah. Okay, I've got an advert with a guy coming out of his car. All the adverts I got on YouTube, they're always old guys, right? Because they know how old I am. They think I want to buy something like another old guy. Okay, that's it. Yeah, so we've got a couple of minutes on this. So here's your SARS-CoV-2 particle waving its spike glycoproteins around like trees in the Kieran storm. Okay, and on the inside you've got the genome wrapped around the heliconuclear capsid. So the first step is binding to the ACE2 receptor, which I believe is going to be this yellow thing. Oh, I'm wrong, it was the purple thing. So ACE2 is purple. Okay, so you've got these receptor binding domains that open up, they bind to ACE2. And the next thing that happens is a co-receptor, which is this protein here called TMPRSS2. It's a protease, and it will cleave the spike protein at two sites and kind of detach this S1 domain and just leaving this S2 domain. This is the fusion domain of the glycoprotein. So once this has happened, then the fusion domain is going to elongate. It's going to project these hydrophobic peptides into the host cell plasma membrane. So they kind of get into the host cell plasma membrane, and then the whole thing collapses. And this collapse is going to bring together the envelope of the virus and the host cell membrane, and then the virus kind of just dissolves into the plasma membrane. And then whatever was inside the virus particle is now transported into the cell. Okay, so that's how SARS-CoV-2 gets its RNA into the host cell. And what's going to happen afterwards? I think I'll probably skip this one. Yeah, maybe not. So next thing that's going to happen is you have virus genes that are expressed. So if you look at the sequence of a coronavirus genome, there are always these two long open reading frames, ORF1A and ORF1B. Then you have, in the 3' part of the genome, different open reading frames, and they definitely have got to code for the 4 structural proteins, spike, nuclear capsid, the M and the E proteins. And then there are a bunch of different open reading frames that can be variable from one coronavirus to another. Now, this is a pretty big genome. It's kind of the longest genome for an RNA virus. So it's a complicated structure, lots of different genes in there doing different things. So basically what's going to happen is once the virus RNA gets into cells, the host cell ribosome will bind here at the 5' extremity, and it will start to translate these two big open reading frames into polyproteins called PP1A and PP1AB. And in amongst these polyproteins, you've got these protease genes, which will cut down all of these polyproteins into the constituent functional virus proteins. Another thing that is here is the RDRP, RNA-dependent RNA polymerase, and this is what's going to synthesize the negative strand, okay? And then also smaller negative strands, and then from the negative strands, you get all the other mRNAs that are synthesized. So you've got these two enzymes essential for the virus replication. RDRP, the RNA polymerase, and the two different proteases. And in fact, some of the drugs that are effective against SARS-CoV-2 are inhibitors of these two enzymes. Okay, so what about pathology? If you catch one of these viruses, man, I'm going to be so late. Probably going to have to skip a lot of this. Let me just check, because I have quite a lot of things to say, and I'm not going to be able to do this. Yeah, I'm going to switch. I'm going to skip a couple. I'm just going to go to SARS-CoV-2 pathology. And I'll do this and I'll miss SARS out, okay? Right, so seasonal human coronaviruses. Normally, it's not going to cause anything that is problematic, okay? Most people catch these viruses several times in your lifetime, and they'll give you a kind of just a mild cold. But they can cause pneumonia, okay? And the population that's at risk for a severe form are children under 2 years old, elderly patients. And in that case, you have some indication that the virus is replicating in the lung, because you'll have wheezing, you know, if the doctor listens to your lungs and they'll hear something's wrong, more people have fever. So, you know, these viruses, they're generally harmless, but still can cause serious infections in some at-risk populations. Now, if we look at SARS-CoV-2, what's going to happen is that the incubation phase is about 5 to 6 days between infection and symptoms. The first symptoms, and this was for, you know, the first wave in 2020. Fever, cough, fatigue, nothing very specific. The only thing that's really specific was this loss of smell and taste. And then after you've got the initial symptoms that appear, one of two things happen. Either you feel bad for a few days and then it gets better without really needing to go and seek medical attention, or things will get progressively worse. Okay, and then after about a week of these first symptoms, then the people who have a severe form, they have difficulty breathing, and then people who have a really severe form will find that they'll need to be hospitalized, they'll need oxygen, and then at the end can even die. So what is happening is that the first phase of the symptoms, this is what's called the viral phase. This is a kind of direct result of virus replication. Whereas in this secondary phase here, where you have the severe effects and progressively get worse, this is to do with the host inflammatory response in the lungs. And that means that these antiviral therapies, they work pretty well if you give them quickly, early, but later on, not so good. Okay. Now, how dangerous was SARS-CoV-2? Now, how much of a menace was it? So in French, the best stats were published in the summer of 2020. And this was a group of statisticians at the Pasteur Institute who managed to break down all the available data to calculate the infection fatality ratio for each age group. So that means, this number means, if you are infected, what is the risk of you dying? Okay, so for young adults, 20 to 29 years old, it's less than 1 in 10,000 risk of dying. And it got progressively higher. Okay, so if you're like if you're over 70 years old, you had about a 2% risk of dying here. And overall in the French population, this was about 0.5%. So for a virus that was going to infect everyone in a huge epidemic, could have had about 250,000 deaths in 2020. Okay, so if you look at the data, and I haven't like brought this up to date because the message is just the same. Up here, these are the number of cases per week or per day actually, and this is the 7-day average in the blue line. So you've got these different peaks of infection, and then this is the number of daily deaths. Okay, so in the first peak, you've got a load of deaths here, not very many infections. Well, that's because we didn't have the testing kits available. So in fact, the real number of infections was way higher than this, okay? Then in the second phase, you've got good testing and still quite a large number of deaths. And then when you notice when we get to, 2021, 2022, we had these massive peaks of number of infections, but the number of deaths stayed pretty low, okay? So in more recent waves of SARS-CoV-2, the death rates have been much lower. So what has changed? Okay, two things have changed here. One of them is the population has changed. At the beginning, this was a new virus for everybody. But by 2022, either everyone had been vaccinated or had already been infected. So the virus was infecting a population that already had some kind of protection. So these stats are not available anymore, or can't be found that easily because the website has changed. So they showed here the number of positive tests per million and the number of entries into intensive care per million people amongst the vaccinated population, which is green, and the non-vaccinated people, which is in blue. Okay, so up till the winter 2021-22, the vaccine had about a 50% protection against infection, but it was pretty effective against critical illness. So it was working to protect people from severe infection and deaths. Now that was true up until the end of 2021-22, but then later on in other ways, what you found was that later on in 2022, there was almost no difference in infection rate between vaccinated and unvaccinated people. There was still about 60% protection against severe forms and death. So this is one of the things that was bringing down the death rate. The population that the virus was infecting was different. Most people were vaccinated, the death rate was way lower in the vaccinated population. Now, the other thing that was changing over time was the virus. Okay, so I don't know if any of you have followed this, but over the last few years, we've had waves of different variants of SARS-CoV-2. So up until end of 2020, it was like the first wave. And then we got these different variants, Alpha, Delta, and then all of these Omicron variants here. And then the recent ones, 2023, it's XBB. So each new wave has replaced the previous virus population. And the reason this is happening is because if you look at people who've been vaccinated and the ability of their blood to neutralize the virus, so in the gray dots, that's the wild type virus, okay? The higher the scale is up here, the more active the serum is to neutralize the virus. So vaccinated people were protected from the wild type virus, a little bit protected against Delta. Omicron, nothing, okay? And it was also true for people who had previously been infected. So these were data from Olivier Schwartz's group in Pasteur, published at the beginning of last year. So that's why we had like a huge wave of Omicron infection in 2022. So I got it. I was one of these vaccinated people who had no protection against Omicron infection last November. Now, what is happening here? Okay, so if you have a little bit of a look at the S protein trimer, you have two domains, S1 and S2. S1 is the receptor binding subunit, and particularly this C-terminal domain is the receptor binding domain. Now, this is also data from the paper. What they looked at was in So this is the 3D structure of the receptor binding domain. And in red, you've got the amino acids that bind to the receptor, ACE2. And in red, you've got the mutations in the Omicron virus. And these kind of like circles, these are the regions that are targeted by antibodies. If you have an antibody that binds here, the virus can't infect a host cell. So what they show is that, in fact, all of these mutations that have accumulated in Omicron, where they stop the antibodies from binding to the RBD, but the RBD could still bind to the receptor. So it could still infect people. Now, the other thing that happened, was kind of interesting, but I'll skip this a little bit, is that because the spike glycoprotein changed, it was pushed to change by becoming more resistant to antibodies, the properties of the virus changed a little bit. So this is an experimental infection in hamsters with different types of virus. And just looking at this curve here, this is the weight loss of the hamster, okay? So gray is wild type virus. Orange is the Delta variant. So after these animals got infected, they got sick, they lost weight. But with Omicron, they didn't lose weight so much. Okay, so the virus was less pathogenic, and they saw that there was less replication of the virus in the lungs. So in fact, what had happened was that Omicron had become a little bit more like a seasonal coronavirus, more infectious, less pathogenic, and it's because it would switch to the entry pathway. So SARS-CoV-2 2020 was using this TMPRSS2 as a co-receptor and with cell surface entry. But Omicron was internalized and used the endosomal pathway, doing the same kind of thing as a seasonal coronavirus. So the nature of the virus changed, and that meant it became, in fact, less pathogenic. So compared to Delta variant, you had less risk of hospitalization if you catch Omicron and less risk of having a severe infection, even if you had to be in hospital. And that's also, adjusted by vaccination status. So, okay, the host population changed and the virus changed. And that's why we've had fewer deaths in the last couple of years. And XBB seems to have stayed about the same. Okay, it hasn't really become more pathogenic. Okay, maybe I'm on the last slide here. Okay, so what's going to happen in the future with these variants? Now, this is a slide from a paper just published a couple of months ago, and they compared the mutation rates in different proteins of different endemic human viruses. So if you remember last year in mechanisms of evolution, NA over NS, the non-synonymous versus synonymous ratio, they looked at different viruses to see where you get the accumulation of more non-synonymous mutations. So influenza type A, the hemagglutinin protein and the neuraminidase protein, they accumulate mutations over time, 6 per year. The influenza type C doesn't seem to do this. You've got 2 negative strain RNA viruses, and then these endemic coronaviruses. One of them is carrying on mutating, and the other one is not mutating, NL63. So I think what is going to happen for the future of SARS-CoV-2 is going to be one of these two. But it's rather too early to really predict what's going to happen. We don't really know whether it's going to stabilize like this one or whether it's going to carry on mutating. And so far, the mutation rates that were in this paper for SARS-CoV-2, they're like 18 or 20 on this scale. So it's really fast compared to all the other endemic human viruses. Now, one of the interesting things about this is that NL63, if you remember, it uses the same receptor. It uses ACE2 as the membrane receptor. And you think, well, why isn't this thing mutating just like SARS-CoV-2? One of the reasons might be that somehow the way the S1, the receptor binding domain here, is constituted means that it doesn't really have much space to mutate. If it mutates, it won't be able to bind anymore. So I, this morning, emailed somebody, Jesse Bloom at the Fred Hutchinson who's one of the people that's done a lot of great work on this. I'll use a couple of minutes to tell you about this because it's fantastic. So what his team has developed are what's called deep mutational scanning. That is, he's got a way to mutate every single amino acid in the whole of this receptor binding domain, express them on a bank of a library of yeast cells with the different RBD mutants on the outside, and they can test whether they can still bind to the ACE2 protein. So for each mutation, you can find out whether it's still going to function or not. So I wrote to him very quickly this morning, said, oh look, have you tried this one to see whether it can mutate and still work? And he said, yeah, well, we're thinking about it, but we haven't done it yet. So when this is done, we'll be able to find out if there's really a fundamental difference between this spike glycoprotein and the SARS-CoV-2 that could maybe give us an idea what's going to be the future for SARS-CoV-2 evolution. So I'll skip my previous predictions from previous years when I've done this lecture. So what's the situation we have now, okay? So my prediction from last year was, are we going to have a post-Omicron variant? Yes, that happened. So Omicron was replaced by the XBB variants. I also was predicting a big winter influenza epidemic last year because I thought, you know, we were in line for that. Well, it wasn't really that big. Okay, so this is the data from Santé Publique France. So that's last year's influenza epidemic compared to previous years. Started a bit earlier and lasted a bit longer, but it's not like it wasn't massive. And my predictions for 2024, I think what's going to happen is that we should probably get a combined COVID and flu vaccine. But if this is going to work, then the COVID booster has got to become more efficient, because the booster vaccination didn't really help to generate specific immunity against Omicron or the other variants. So there's a big issue in immunology and vaccine development that will need to be solved in order to get more effective vaccines against future variants. Okay, so that's time up for today, and I'll just go on to microbe of the week. Okay, so we've run out of time and get the results here. Okay, so the last question was, why would a listeria infection be more dangerous for a pregnant woman than a salmonella infection? And then how does listeria invade cells? And what's the unusual product that can be sprayed on meat to prevent contamination with Listeria monocytogenes? So the answers were, okay, just so everybody's clear on this. Listeria is more dangerous because it can cross the placenta and infect the fetus and cause infection or death of the fetus. Salmonella can't do this. How does the virus get into cells? Well, kind of 1 point for the process, 1 point for the virulence factors on this one. So it induces its own internalization into cells. It escapes from the endolysosome, and then it can move around inside the cell and from cell to cell by polymerizing actin. So it's kind of producing a kind of trail behind it of polymerized actin that pushes the bacterium along in the cell. So the virulence factors are internalins for attachment and internalization. Listeria lies in O to escape from the endolysosome, and the ACT-A protein for the actin polymerization. And last question was, unusual product on meat to prevent listeria contamination? Bacteriophage. There are commercialized mixes of bacteriophage which can be sprayed. I think not available in France, right, but probably in the States. Spray these bacteriophage on the meat product, and any listeria that lands there will get killed and won't grow. It's all a big problem for listeria because it's psychrophilic. Okay, carries on growing in the fridge. That's why listeria contamination is a big deal for like these meat products. So got a few good answers, some answers not entirely complete. So basically the end of the result is we got this. So I think we got in equal first place, we got Keridwen and Solène. And in third place, we got Alec. So, Kerrigan, Solène, can you come up, please? Only one? No. Someone's not here? Oh, well, unlucky for her, because in that case, you don't have to share first prize. Okay, so here you go, first prize. Fermentation products. There you go. Well done. Normally with two. Okay, so if you could get Soleil, you should share that with her, equal first place. But you know, she should have turned up, right? Second prize, third prize, another fermentation product for Alec. Here you go. Thank you very much. Please enjoy. Okay, that's it. So I guess we're on time. And any questions before I run away? Well, I'll hang around.